Thank you, Randy. Well, you may not have wanted me to be the first speaker, but it is truly an honor to be the inaugural speaker for this. So, yeah, so I think Randy kind of hit it on the head. I think I've had this interest for a lot of years, is trying to understand, you know, if there are traits or if there are phenotypes out there that are, you know, highly heritable and fairly common, at least common with respect to a lot of the disorders that we know are deleterious, like, you know, achondroplasia or Apert syndrome, and these kind of things that are, you know, kind of 1 in 100,000 frequencies. We're talking about things that are, you know, 10, 100, 1,000 times more frequent, more common than that, and yet they're still highly heritable. And so the question that motivated a lot of this research I'm going to talk about is, you know, why are those genes still hanging out in the population? And the in particular, well, I guess the phenotype that I'm going to be looking at is schizophrenia for this talk. So I just had a cold, so if my voice starts going out and I leave the room, it's not because anything you guys are doing or looking at me badly or anything. It's because my voice, and it happens every time I get a cold, I get laryngitis. But I think that I'm going to be just fine, so we'll see how that goes. So, you know, I think this bigger question about the evolution of the evolutionary genetics of these psychiatric disorders plays into some of the central questions in genetics right now. So one of the big questions is, you know, do the genetic variants or the alleles, we call these risk alleles, do they tend to be recessive? Do they tend to be additive? Do they tend to be dominant? Is there a bias towards one or the other of these? And I'll talk about what those mean in just a second. I think it's a pretty mixed audience, so if, you know, and feel free to stop me if there are questions for clarification. I'd be happy to take those as we go. You know, another kind of long-standing debate in genetics is, are there just a few risk alleles of large effect, or are there a lot of risk alleles, hundreds or maybe even thousands of different alleles, or tens of thousands of alleles, each of which confer only a very, very small proportion of variance to the total phenotypic variance that you see. And do those alleles that exist out there, they tend to be common, they tend to be shared by lots of people, lots of unaffected people, and it's just, people who have, you know, these disorders have too many of those risk alleles, or do they tend to be rare and fairly family specific? That's what I mean by a rare allele. So typically the cutoff that we use in genetics for a rare allele is where the minor allele, the least frequent allele, has a frequency less than 1%. So that's kind of the arbitrary threshold I'll use for rare alleles. And finally, do those alleles tend to be shared across ethnicities, or they tend to be unique within ethnicities? So in a sense, this question, number 4, can be seen as a, you know, kind of a part of number 3. If the alleles tend to be rare and shared within families, then certainly they're not going to be shared across ethnicities. So 4 is really kind of a subset of 3. So these issues here really concern, I guess you can't see it on the TV, can you? That's interesting. Huh. That's the angle. Can you guys see it? No? Oh, it's fine. I don't mean it, so. Anyway, so I think it's fine. So, you know, yeah, exactly. Old school, so I'll point to things on the screen. So, you know, these issues concern what is called the genetic architecture of traits. And there's practical importance to these questions as well. I mean, to the degree that the risk alleles aren't additive, they have small effects, and they're rare, then finding those risk alleles is going to be very difficult. And therefore, the promise of personalized genomic medicine is just a promise. I mean, it's still fairly distant. If the risk alleles, on the other hand, are specific, and another, I think, issue, a side issue, is that if the risk alleles are specific to ethnicities, then there's a risk that the genetic discoveries that are being made today have a Eurocentric bias. So as I said, the phenotype that I'm going to be focusing on here is schizophrenia. Probably a lot of people already know what schizophrenia is. It's highly heritable. About 80% of the differences of the variation in the population in propensity to develop schizophrenia appears to be due to differences between people and the alleles that they have. In other words, about 80% heritable. It affects, depending on really the culture, so there is some variation across cultures, but it's within a fairly narrow bound. Between about 0.5% to 1% of people worldwide will at some point in their life will have symptoms that would be diagnosable as schizophrenia. It is observed across every culture that's been studied. I don't think there's been any studies on hunter-gatherers, but I would be surprised if it's not also in hunter-gatherer societies. As I said, the heritability is about 80%. And molecular genetic studies have, I think, recently begun to shed a lot of light on the genetic architecture of schizophrenia, those basic questions that we've come out. I think people outside of genetics have kind of come to one of two different kinds of conclusions about what's going on in genetics. Either they are way over-optimistic and think that we've learned a lot more than we have, or if they have a little more knowledge, they think that it's been a total wash and that we haven't found actually anything. But I think both of those both extreme views are not right, and I hope to kind of show you that the middle road, that we actually have learned quite a lot about the broad genetic architecture of schizophrenia, even though it is true that we haven't done a very good job of identifying specific alleles that confer very much variation. And that's just probably because that's how nature built it, or more to say, or more to the point, how evolution built the system. So the genetic architecture of any trait is due to the evolutionary forces that acted on those traits. And we're talking about forces that are anywhere from hundreds of years ago to many millions of years ago. So in other words, the current genetic architecture of traits is due to things that happened in the past. And if there are evolutionary geneticists in the room, they'll probably recoil at my simplistic way that I'm going to describe the two kinds of ways that this could work. But I think for the most part, I think it's going to be okay for this audience, and that is, or for this talk, I should say. And that is one way that you can think about the evolutionary genetics of risk alleles is that they've been neutral with respect to fitness. There's two kind of sub-ways this could work. One is that the risk alleles just simply had no effect on fitness at all. Perhaps the phenotype had no effect on fitness historically, or perhaps those risk alleles didn't confer a risk historically, and it's just something in the modern environment, a gene-environment interaction that's had those risk alleles change and become risk alleles only in modern environments. And the second is that the negative fitness effects of these risk alleles are countervailed or balanced by some positive effect that perhaps isn't perfectly obvious. So this second one is often called balancing selection, and that's why I say that evolutionary geneticists might recoil at this. I think usually people talk about neutrality, they talk about balancing selection, and then they talk about negative selection or purifying selection, which is the next one I'll talk about. The other, I think, but I do think I should say that in both cases, right, even if you're under a balancing selection, it is the case that the alleles have the same, on average, have the same marginal fitness effect. And the only difference between these two is that under balancing selection, there's some homeostatic mechanism that ensures that the allele isn't going to go extinct. Whereas under neutral evolution, it's purely up to chance. And these things can drift up to high frequencies or low frequencies, but it's purely governed by chance, kind of a Brownian motion of genetics. The second broad class of forces that may have acted on these alleles is what you could term negative selection or more technically purifying selection. And that's that simply the risk alleles are simply deleterious. They're simply negative, and they're being driven out of the population. But the issue is that there's so many ways, there's so many possible sites that these mutations, that there's so many genes that affect this phenotype that there's a huge mutational target size. And because of that, even though each individual allele is being driven to extinction, albeit slowly, there's a constant replenishment of new mutations in the population. And that leads to what's called the mutation selection balance. At any given time, there's a balance between new mutations that are rising and old mutations that are going extinct. And so all populations, we know this happens, all populations are awash with deleterious mutations. It's just a matter of how many there are. And so one idea is a lot of these psychiatric disorders are just ramifications of that deleterious mutational load that affects the brain. Some people ask, well, where's positive selection? How does that fit in here? Well, it probably does fit into some degree. I mean, certainly as alleles are fixating and being driven to fixation, that will create transient genetic variants. But for the most part, most evolutionary geneticists don't really focus on positive selection because of that, because it does lead to transient and not balanced genetic variation. And so I also won't focus on positive selection. I'll really talk about these two. So the goals of this talk, I'd like to discuss some research that I've done or that we've done in my lab that I think sheds some light on these basic questions about the genetic architecture of schizophrenia. And then pivot towards, just briefly, well, I guess in each of the sections I'll do this, as pivot towards what that genetic architecture may tell us about the forces that acted on schizophrenia risk alleles over evolutionary time. So here's the outline. So I'll first try to get some leverage on the question of whether schizophrenia risk alleles are recessive or more recessive than expected by chance. Then ask whether there are just a few schizophrenia risk alleles of large effect or if there are many of small effect. I'll give you a hint. It's definitely the latter. And I'll show you evidence for that. Third, do schizophrenia risk alleles tend to be common or rare? And again, a hint is that it's both, and some evidence for that. And then, are schizophrenia risk alleles shared across ethnicities or unique to ethnicities? And that's some work that a graduate student of mine has done. And I'll talk about that. And if I have time, I'll talk a little bit about some future work that we're working on now. So first of all, are schizophrenia risk alleles more recessive than expected by chance? So this relates to the concept of inbreeding. So everybody knows what inbreeding is, right? Brother, sister mating, cousins mating, something like that. It tends to cause revulsion, right? Probably for good evolutionary reasons. But the fact is that everybody in this room is inbred. You all are inbred, and it's just a matter of degree. And the reason that has to be true is because your parents are related to one another. Your parents share common ancestors. It's a matter of how far back you have to go before you find that first ancestor. If you're a cousin, you only have to go back one generation. If you're second cousins, 2 generations. But all of us are cousins in this room. We might be 20th cousins or 40th cousins. In fact, we're cousins many, many ways related. I might be 8th, 15th, 21st, 32nd cousins with Randy and so forth. And what that does is that creates individual variation in all of us in the degree to which we're inbred. And basically what I'm going to show in this part of the talk is just relating that variation and how much we're inbred. In particular, we're measuring this at the genetic level to the propensity to developing schizophrenia along with some other traits that we've looked at. So in our lab, we've measured levels of very, very distant inbreeding. As I talked about, this being like second cousin all the way back to 50th or even 100th cousin kind of inbreeding. So where the common ancestor could have lived 100 generations ago. In non-selected samples using single nucleotide polymorphism chips. So we call that SNP. Stands for single nucleotide polymorphism. I'll explain what those are in just a second. I'll actually explain it right now. So SNPs, so SNPs are single nucleotide polymorphisms. This is where so in the genome, right? This is where there's a particular base. So here's person 1. That's this is the, I just lost the word for it, or the homolog that they get from the mother, the one from the dad, and so forth. So this is person 2 from mom, haplotype was the word I was looking for. Person 3 and person 4 and so forth. So if you look across the genome at this first site, you see that some people actually have a G there, some people have a T, and some people have 2 Ts, 2 Gs, so forth. Okay. There's a lot of sites, actually the majority of sites, if you look in your genome, that are going to be homozygous, and if you look at a lot of people at that site, everybody's going to have the exact same allele. We call those monomorphic sites, and those are there. But these other sites are polymorphic. So kind of by definition, we call a SNP a place where there's a polymorphism where the rarest allele has a frequency greater than 1%. The minor allele frequency is greater than 1%. And pretty much on a SNP chip, there are always exactly 2 alleles that we're looking at, a T and a G, or a C and an A. Certainly, there are some 3 SNP alleles out there. Those aren't measured on SNP chips. They tend to be very rare. There might be only a few thousand of those. I'm aware of no 4 allele SNPs that exist. So there are about 15 million of these positions across the genome. 10 or 15 million. Only about 1/10 of those are actually measured on any given SNP chip, any given SNP panel. And that's okay, right? We still get, even though we only measure 1/10 of all the polymorphisms out there, because there are statistical associations between this SNP and this SNP. In other words, if you have an A here, it's probably likely you have a G here. That's called linkage disequilibrium. Because of LD, we can do a really good job predicting the other 9/10 of those that are left off with just the 1/10 that we've measured. 1/10 that we have measured. So really, with SNP chips, we're getting a pretty good canvas of what is actually in the genome that is common, that it's over 1% in the population. Sites that don't vary are ignored on SNP chips. And then occasionally you'll get, a rare mutation, say only one person in 10,000 has a T at this position. That also is going to be ignored on SNP chips. So SNP chips don't measure things less than 1%. Finally, some last bit of, I think most people probably know this, but just for terminology for people who don't, if you've got a C from dad and a C from mom, you'd be homozygous at that site. If you have a C from mom and a T from dad, you'd be heterozygous at that site. That's a heterozygous mutation. Most mutations, by virtue of them being rare, are heterozygous. Pretty much the only time that a very rare mutation is homozygous is when there's inbreeding. And then homozygous mutation here, so 2As, that probably comes from the same ancestor. And really the goal of this part of the study, of this study, was to measure these rare homozygous mutations along with other homozygous common variants that exist in the genome. So why do we care about homozygous mutations? So I think most people are aware that genes come in, the effects of genes come in three kind of flavors. Remember this from first year biology. You have recessive effects, additive effects, and dominant effects. If the T is recessive here, the effect of a TC is going to be exactly the same as the effect of a CC. If T is additive, the effect of TC will be midway between the effect of T and C, exactly midway. And if T is dominant, the effect of TC, it's going to look just like a TT. Okay? But I should say, though, that even though you've learned this in biology, they come in three flavors. They don't come in just three flavors. They come in an infinite number of flavors. It's really a rainbow that goes from perfectly recessive to partially recessive, all the way to additive, to partially dominant, to fully dominant, to even over-dominant. Okay? So there's a whole range of which these effects can occur. And so it's simplistic to think about just a recessive, additive, or dominant. But nevertheless, what we're going to be talking about are mutations that have been biased towards this end of the spectrum, towards being more recessive than would be expected by chance. So selection keeps all variants that affect fitness rare, right? Under purifying selection, I should say, all variants that are harmful to selection are kept rare. In other words, they're mutations. It's probably a little too strong to say all, but let's say the vast majority of. However, purifying selection is going to remove the dominant mutations, the mutations at this end of the spectrum, the ones that are most additive and most dominant, more quickly than it's going to remove the recessive ones, the ones on that end of the spectrum. Okay? And because of that, the current mutations, the ones that are existing right now, the ones that have kind of passed through the gauntlet of selection for tens and hundreds of thousands of generations, tend to be biased towards being more likely to be recessive. Okay? In other words, right, variants that affect fitness that have been under purifying selection tend to be biased towards being more recessive than would be expected by chance. And because of that fact, homozygosity of fitness affecting mutations is particularly deleterious. So here's a cartoon of kind of very simple inbreeding. This would be brother-sister inbreeding in this case. So you've got mom here with her two haplotypes, and dad with his two haplotypes. They mate and they have two offspring, say a boy and a girl. If those boy and girl mate, then the child here is going to have tracks of their genome that are purely homozygous. Okay, they're called identical by descent homozygous tracks. In this case, if you look closely, you can see that actually there's 3 of those. There's one that comes from this right here, that comes from that haplotype from dad. There's one long one here, the yellow, that comes from right about there from the mom's first haplotype, and then a smaller one there that comes from the mom's second haplotype. In other words, inbreeding leads to these extended tracks of homozygosity. So it's not just homozygosity at particular alleles, it's homozygosity at entire big chunks of the genome that are getting passed down. There's only about 33 meioses or something that occur at any given reproduction. And so over time, those chunks will get smaller, right? So if your common ancestor between 2 people was 100 generations ago, those chunks are going to be very small, say about a centimorgan long, 1/100th of a morgan long. Whereas if it's just from brother-sister, as in this case, on average, those are going to be very long, about 50 centimorgans long. So the percent of the genome, if we count these up, that is in those homozygous tracks, we can call that F. F is the inbreeding coefficient. In particular, because we're measuring runs of homozygosity here, we can call it FROH, F rho. I think what's critical here to understand is that in those homozygous chunks, it's not just the measured SNPs, the common stuff that's homozygous, right? It's everything in between there that's also homozygous, including the rare mutations. So even though we've measured just the SNPs to get to this point, to figure out whether you're in these homozygous chunks, everything that we haven't been able to measure, all these mutations that don't exist on SNP chips, we can still infer, right, that those rare mutations that we haven't been able to measure, we can still infer that they're homozygous in those regions. All right, so that was just a bit of prologue to what's coming. So inbreeding via increased homozygosity reveals the full effect of recessive fitness-affecting mutations. Inbreeding only is deleterious after the selective pressure has biased mutations to be more recessive than would be expected by chance. And I feel like this is a lot of repeating myself, but therefore deleterious inbreeding effects are evidence that risk alleles are more recessive than expected by chance, and also evidence for selection against those risk alleles. And so what we've done in our lab is measure individual differences, as I said, I think in the very beginning, between people in very distant levels of inbreeding using SNPs, using SNP chips. And so how do we do this? So here's an example of a SNP chip. We've measured the T here. There's, on average about 5,000 or 6,000 base pairs between these SNPs. So, maybe several thousand base pairs before we measure our next SNP. Looks like a few thousand there before our next one, a few hundred, and so forth, right? So there's big gaps between these SNPs. But if we see a long run of SNPs that are all identical within a person, right? So for this person, they're T here, but they're also T here. In other words, they're homozygous, homozygous, homozygous. There's a long run of homozygosity there. What we can infer, right, and then the longer that gets, the more likely this is the case. What we can infer is that that haplotype came from the same ancestor as that haplotype. These, in other words, are identical by descent. And because of that, everything between those two guys, right, everything between these SNPs that we've measured is also probably identical. Unless there was a mutation since the last common ancestor, everything on those two identical by descent haplotypes is going to be identical, including those unmeasured rare mutations. So we've taken this approach, these runs of homozygosity using whole genome SNP data, and we've, we did some work to figure out, I think I've got that here, yep, this is my graduate student, to figure out, optimize the best ways of measuring this. It's not a simple problem because even though you'd think, 65 homozygous SNPs in a row wouldn't happen much by chance, it actually can because of the LD I was talking about earlier. So it took some work to figure out what kind of the optimal method was to optimize sensitivity and specificity for finding these runs of homozygosity. And we did some other work to suggest that because in outbred populations, in unselected populations or unselected samples, the level of inbreeding is so low, the variation in inbreeding is tiny. Most people just have very, very small amounts of inbreeding. And because of that, it requires enormous samples to be able to see any signal. And so we're talking upwards of 20,000 and above just to get to kind of 80% power, statistical power to detect any effect that we might expect. So we need large samples to be able to do this, large samples measured on whole genome data. Luckily, such samples do exist. So the first foray into this, we use the Psychiatric Genomics Consortium data. It's called PGC1. There's now a PGC2, which I'll talk about in a second. This is comprised of 17 studies, 10 countries. They're all Caucasian, and about a total of about 21,000 individuals. And the prediction was simply that when we tally up these runs of homozygosity within each person, right? So every person has a sum that's converted to a percentage, and we regress the probability of being schizophrenic on that percentage. The prediction is simply that these runs of homozygosity are going to predict a greater likelihood of schizophrenia. And that is indeed what we found, which was published in 2012, I think. So what we're seeing here is each of those 17 studies is on the x-axis. So on the y-axis is the estimated change in odds. So I've converted it from log odds to just change in direct odds of for each 1% increase in FROH, how that changes your odds of getting schizophrenia. So if there's no change, it would be 1, right? As you increase, it doesn't change the odds at all. If the odds were 2, that would mean it would double your risk of having schizophrenia. And then these lines around here are the 95% confidence intervals for each of those studies. Okay? So the first thing that jumps out at you is if you look across here, right, each of those lines intersects this line of 1, right, which means individually none of those studies were statistically significant. So it takes a lot larger samples than any of those individual studies to find this effect. But we used the mixed linear effect model, right, to treat data set as a random factor. And basically it's kind of a meta and analytic approach. Looked at the overall effect of runs of homozygosity on all 22,000 individuals. And the effect there is highly significant. About 1 in 1,000 would the null, if the null were true, this would occur about 1 in 1,000 times, this size effect or larger. So in other words, it's unlikely that this happened by chance, although obviously we can't know for sure. And we are trying to replicate that now, and that's a whole other issue which has gotten complicated. But basically what that effect says is that the odds of schizophrenia increase about 17% for every 1% increase in FROH. So it's not a huge effect. It probably explains about 0.25% of the variation of schizophrenia risk. So in other words, this isn't a smoking gun, right? Nothing in genetics so far for schizophrenia has been a smoking gun, but it does appear to be related. But I think there's a more important factor, right, that this is suggesting, which I'll get to in just a second, that tells us something about the evolution of, at least the evolution of schizophrenia risk alleles, regardless of the effect size. We've investigated this on several other phenotypes. We just now have a manuscript going out on IQ. We see the expected relationship that it does not achieve statistical significance, which once again tells you that the sample sizes need to be really huge because the effect size that we're seeing is exactly what we expected going in, but we're doing it on 8,500 people. So we probably need to get up around 20,000 to see a significant effect. This one surprised me. This was one done by colleagues of mine in Australia. But basically, if you code personality traits according to whether they're more or less desirable, this is only a sample of 8,000 people, so it's even smaller than the one that we did for IQ. What you see is a significant association with runs of homozygosity with increased harm avoidance and with decreased novelty seeking and reward dependence. So basically, the personality traits that are that people desire the most in a friend or a romantic partner, those also seem to appear to show inbreeding depression, which I was skeptical of this going in, but the effect sizes or the significance of those were kind of convinced me that probably is the case. We looked at this in major depression. We did not see any relationship at all. Actually, we saw a relationship, but it was extremely confusing. We don't know how to explain it. It differed completely depending on whether it was measured on an Illumina chip or an Affy chip. So it makes no sense. We don't know what's going on. But we did publish that and just kind of said, look, we really don't know what's happening here. And we're now in the process of trying to replicate this in a much larger schizophrenia sample, the PGC2 sample. And so it's not independent of that original 21,000 sample. So this is about 60,000 independent people. And that 60,000 independent sample, this is a graduate student who's doing this now, we've not been able to replicate our original result, which I find extremely surprising. There's a huge caveat here. I found out only like a week ago that the way she'd been doing it was basically wrong. It's a first-year grad student, so it's not her fault. It's my fault for not being a better mentor and spending more time with her. So I think it's on hold at this point. So, but there was about a month this summer where I thought, holy cow, I cannot believe that didn't replicate. It's a one, I mean, we didn't do any fishing originally in the data. 1 in 1,000 p-value. You'd think something like that would certainly replicate in a much higher powered sample. So I think the verdict's, the verdict's out. Or wait, is that the right way to say it? The jury's out. Jury's out. The verdict isn't in. The jury's out. So we'll see soon. Hopefully we'll know that answer soon. Okay, so in summary, this first part, are risk alleles more recessive than expected by chance? I think the answer there for schizophrenia and for some of these personality traits that we saw is yes, at least currently. The jury's out, as I said. But FROH is significantly related to risk for schizophrenia and other traits potentially related to fitness as well. The importance of that isn't so much in the amount of variation that we explain in schizophrenia or personality. The importance, I think, really is that it points to the fact that these schizophrenia risk alleles have been under negative or purifying, on average, purifying selection over evolutionary time. Because there's really no other good explanation for why there would be a bias towards recessivity on those risk alleles, as I explained earlier on. And so what I think that implies is that the schizophrenia risk alleles that over evolutionary time were most dominant or most, on the dominant end of the spectrum or additive have been removed. But why would they be removed, right? The answer is obvious, because they've been under negative selection. Selection has been removing those. So, as I said, that suggests that schizophrenia risk alleles have been under purifying selection across human evolution. Okay, so the second part, this one definitely takes the longest, and that's just because there was a lot of kind of preamble I had to get through with.
A lot of people have proposed that these alleles actually give a selective advantage somehow or another. Wrong.
Well, I think it's definitely wrong on average, but I think that was the prevailing notion in kind of evolutionary medicine, really, for a long time, is that if something is highly heritable and it's kind of hanging out in the population at frequencies of 0.5% or 1%, there must be some reason, and the natural reason that people gravitated towards was balancing selection, frequency-dependent selection or antagonistic pleiotropy, maybe the schizophrenia risk alleles that lead some people to have schizophrenia in other bodies, in other environmental or genetic contexts. They create people who are more creative or more sexy or more intelligent, or these types of ideas. And so there's a balance between the negative effects and the positive effects of those alleles. And that's a kind of a sexy idea. I don't think there's a lot of evidence backing that up. Okay, so are there a few schizophrenia risk alleles of large effect, or are there a whole lot of schizophrenia risk alleles, each of those which have a small effect? So we're starting to get a handle on this question, and I think the answer is fairly unequivocal, to the point where if you're going to argue otherwise, well, I mean, frankly, if you're going to argue otherwise, you're going to get laughed out of a room of geneticists, but I think that the burden of evidence would certainly be on you. So there's now hundreds of genome-wide association studies that have been conducted. So what is a GWAS? We call those GWASs. What are those? Basically, you take 1 million of those SNPs that we talked about earlier, genome-wide, and you do 1 million t-tests. It's as simple as that. It's about as basic of a procedure as you can imagine. Now, of course, there's a multiple testing issue here, right? Anybody who's in statistics knows that if you do 2 tests, your probability of a type 1 error basically doubles. So you've got to correct for that multiple testing comparison. And so they do that in a very simple way as well. They just basically do a Bonferroni correction. They divide it by 1 million. And so the p-value required for genome-wide significance is about 5 times 10 to the negative 8. Right? So it has to be, you have to be really damn sure this thing is associated before you're going to call it. And this is why early on the GWASs weren't very successful, because the standard of evidence was so high. You either had to have a huge sample or you had to have a small sample and get huge hits. The problem is nature doesn't work that way, as I'll talk about. The other issue about GWAS is that they can detect only common variants. So those SNPs that we talked about across the genome, they tend to only predict other SNPs that are also common, that are within the same frequency range. So if you've got a SNP that has an MAF of 10%, in other words, 10% of the haplotypes in the population have a T and 90% have an A, for example. Those types of SNPs are only going to predict other variants, other genetic variants that also have an MAF of around 10%, right? Therefore, right, SNP panels are not going to do a good job predicting things that are rare. They're basically blind to the effects of rare variants, of things that are less than about 1% MAF. So with that, I'm going to talk about two GWASs that have been done on schizophrenia that I've been a part of both of these. I am an author on this. I'm an author of one of about 300 people. So I can't really claim much credit for it. But this was the first one that we did on the sample, the PGC1 sample, about 22,000 people. And we identified 5 total SNPs genome-wide that met that threshold of 5 times 10 to the negative 8. Okay? That actually was a big deal at the time, because in psychiatric genetics, up to this point, we had not found a single thing that was replicating in GWAS. All right? This is now the newest one. This is on the PGC2 on 82,000 people. Now 108 significantly associated loci. This is the Manhattan plot for that, if anybody knows what that is. It's not really worth going into. 75% of those SNPs are in genes, and they're overrepresented with genes that are involved in dopaminergic and glutamatergic and neurotransmission and voltage-gated calcium transmission. So for anybody in biology who knows what those mean, I don't. So, but I've never been to a, so basically everybody at this, I went to the WCPG conference when this was presented that year by Stefan Ripke. And I think, it's about 800 people in the audience when this was presented. I think half the people in the audience were actually authors on this paper. And I've never seen a talk where literally it got a slide got a standing ovation. This slide got a standing ovation. And I think it was just, there's been so much angst in psychiatric genetics about where we were headed and whether it was actually ever going to work. And so I think this is finally proof positive that it can work. It requires huge samples. But there, I'll get to the rest of this in a second. So, so that shows that there's 108 independently significant SNPs genome-wide. If you take the total risk accounted for by those 108, it still only accounts for about 5% of the risk in schizophrenia. Nevertheless, we know that these are real. These are not type 1 errors. We know that because if you hold out one sample and you use it to predict, every one of those predicts in the holdout sample. Okay? And if you do that, if you use all the SNPs with a p-value less than 0.05, so now we've really relaxed it. So we've got a lot in here that are probably truly type 1 errors. But if you use all those SNPs and you build what's called a polygenic risk score, and use that polygenic risk score to predict in another sample, you now predict about 15% of the variance. So now we're starting to get up there. We go from about 0.5% of the variance to 1% to 5%. Now we're at 15%, and I'll talk in a second about how we're up around 25% now. So it's getting closer. So this slide is something that we have in a paper out that that was actually, this is the wrong site. It's Grattan is the first author on that. But this shows the relationship between the effect sizes on the y-axis of SNPs and what are called copy number variants, which I'll talk about in just a second, and their frequency on this x-axis. Okay. So first of all, this is the log 10 of the minor allele frequency. So a -1 would be a 10% minor allele frequency. A -2 would be a 1%. This would be 1 in 1,000, 1 in 10,000. Okay, minor allele frequency. And this is the natural log of the odds ratio. Okay, so I can't even remember what natural log 1 is. It's E, which is 2 point whatever, 3. Okay, so basically that would be about a doubling of the risk right here if you're at 1. Okay, so things less than that would have an odds obviously less than E, whatever E is, 2.3. Okay, so first off, this is that plot for all the SNPs that have been discovered. This is just the PGC. This was intermediate between PGC1 and PGC2. So this is when we had up to like 40 SNPs. And we put onto this plot what we know from copy number variants. So I don't want to go completely into what copy number variants are, but they're basically insertions and deletions. And what's important about them is that we can measure them on SNP chips, but we can actually measure very, very rare instances of these occurring, instances of deletions and insertions and duplications that are very rare, even though they're measured with common SNPs. And I won't get into how we can do that, but it is possible to do that. And so CNVs are important because they were our first insight into the potential importance of rare variants for these disorders. Okay? And so if you plot the MAF of both CNVs and SNPs put together, they're just two classes of genetic variation in the genome. We don't have any SNPs over here, right? Because SNP chips are invisible, are blind to the effects of rare SNPs. They're not on the panel. We don't have any CNVs over here, but that's probably a base rate issue. There's just not very many common CNVs that have been found to affect the phenotype. They're not very common. There's a lot fewer CNVs than there are SNPs. But what the important point is here is there's an inverse relationship between these two. Okay? Right? As the frequency gets lower, right? As the MAF gets lower, you start seeing some big effect sizes individually, right? So, 2.3 to the third power, something like an odds ratio of 15, right? So there are CNVs out there that confer 15-fold risk on schizophrenia. If you have this, you have 15 times more likely chance of having schizophrenia. But it's extremely rare. It only happens about 1 in 10,000 people carry that variant. Okay? You have some other ones out here, right, that confer, about a 4 or 5-fold risk. And those are more common. Now you see that in about 1 in 5,000, 1 in 3,000 people. And then you have SNPs way over here. These are definitely real. These are not type 1 errors, right? And these happen about, this one, for example, about 5% of the population carries that. But it conveys a very tiny effect on risk. Maybe increases your odds by about 5%, right? Not 15,000%, right? Just 5%. Tiny effect on your overall risk. There's an inverse relationship between effect size and allele frequency. What does that say about evolution? Before I get to that, let me say that this line, that's the best fit line. The blue line is the best fit line that explains that data. The red line has nothing to do with the data. That is simply a line defined by a constant heritability of 4/100ths of 1%. 4/100ths of 1%. Okay? And so the heritability is proportionate to 2 times the P, which is the minor allele frequency, times Q, which is 1 minus the minor allele frequency. So the heritability is proportionate to 2PQ times natural log odds squared. Okay? It's not exactly equal to that because there's a constant that you need to multiply by. All right? But if you do this and you say, okay, I want to fit a line that has a constant heritability, that red line is what it looks like. And so what's amazing to me is that red line looks almost exactly like the blue line, which tells us that really there has not been a single variant found for schizophrenia that explains more than 4/100ths of 1% of the variation for schizophrenia. Not one. Or maybe a few. Well, certainly not one that explains more than 6/100ths of 1%. I'll put it that way. I guess there's a few of these, right? That one right there maybe explains a little bit more variation. So what? Okay, so first of all, there's a big gap here that you're wondering about. Well, these are going to be filled in eventually with sequence data. Okay, that will take time, but it will happen. The real question here is, well, why aren't there any variants up there, right? That are both common and that have a big effect. These are the ones that are explaining a lot of variation, right? Because the 2PQ part of the equation is big, and the natural log odds squared part is really big. These are the ones that we were all hoping in genetics we'd find, and we haven't found any of them. And the question is why? I think the likely answer for why there isn't is that is simply natural selection against risk variance. Common variants exist at that end because they have such tiny effects on fitness that they behave basically neutrally. They just drift around in the population. They may eventually go extinct. Hell, they may eventually fixate, right? But it's going to take a long, long time. Those variants are common because they basically confer tiny amounts of risk to schizophrenia, and it took us samples of 80,000 to even be able to tell that they conveyed any risk whatsoever. Okay. What about variants up there, right? These are the C and Vs. These are variants of huge effect, right? 15-fold risk of schizophrenia if you have some of these variants. But those are kept extremely rare by natural selection, and they go extinct quickly. So as a group, those persist because of recurrent mutation. Most of those that you see, they're not exactly the same. They're in the same region, but they're not identical by descent. Yeah.
Seems like the shared distribution should tell you about the fitness cost of schizophrenia.
Yeah.
And if you actually measure fitness costs in large populations, does it match to what you're doing?
That's an interesting, you know what? I think that's an interesting question. I think it also is going to depend on the number of risk alleles that exist. Let me think about that.
So I think that would just fill in the distribution.
Yeah. Yeah.
So on that point, we have 7 minutes left.
Yep.
So if you'd like to take any questions, it's probably best.
Oh, of course.
Wrap in a minute or
Yeah, I'm not thinking through. So let me just finish this through, and then I'll take questions. But I obviously I'd mistimed my talk. But I think this, to me, is actually the most important part of the presentation. I think it's very interesting. So variants at this end are typically rare, but they can drift to low frequencies by chance. Those are also replenished by mutation. And so in summary, are there a few schizophrenia risk alleles or of large effect or many of small effect? It's almost certainly the case that there's a whole lot of small effect. The lowest hanging fruit, the ones with the biggest effect sizes, the one that we have the power to detect, are still only explaining no more than about 4/100ths of 1% of the variation. First of all, that implies that there really are no smoking guns with respect to schizophrenia risk alleles now or probably ever. And I think, secondly, the strongest prediction that purifying selection makes is exactly that, is that there's not going to be any single variant that explains very much heritability for the trait, right? The reason that is because some alleles have, a very large odds ratio, but they have a very small 2pq part of the equation. They're kept at a very low frequency, and therefore the big part of that is canceled out by a very small part of this. If 2 times 0.001 times 0.999, right, you can just throw out the 0.999 as being similar to 1. So this part is going to be really small for a very rare thing, even if that's large. Okay? And vice versa, right? If you have something that's common, if it's under purifying selection, it would never have a big odds ratio if it's under purifying selection. And so I think the strongest prediction from purifying selection is that there's not going to be any alleles that explain very much variation. It's exactly what you see for schizophrenia. So I do think this has some implications for candidate gene studies. There's been hundreds of those published, mostly with positive results, mostly with effect sizes that are 2 orders of magnitude larger than the ones that we've seen with GWAS. I think a likely explanation for this is simply a high false positive rate in the literature. And I think that GWAS is working, but we're not in charge of nature, and there's just a lot of variants out there that each explain a tiny amount of variation. I'm going to actually just talk through the last 2 points here. I don't need to go through the slides, and then we'll
just give us the conclusion.
I'll just
not the logic.
Yeah, let me actually just go to my conclusion side. This is a neat study that my graduate student did. I didn't want time to take about that. But we did find actually that there's a lot of shared similarity for the common SNPs between Africans and Europeans. So I think there's evidence, as I just kind of went through, for purifying selection for alleles that have been under schizophrenia. I didn't talk about some evidence for the total amount of variation due to common variants for schizophrenia. But probably about 1/3 of the total genetic variance of the total heritability of schizophrenia is due to common alleles. It's just that there's a lot of them, even though they have very small effect sizes each. So I think really that's pretty much it. I'd be interested to hear questions from the audience. I apologize about my mismanagement of time,
We
Randy.
didn't tell him how much time. So
Yeah.
Right on the first one.
I learned about it at breakfast, so. I think on my schedule it said an hour and a half, and so I thought it was an hour and a half, but I didn't realize this is the
We're
whole.
gonna fix that next time.
Yeah.
Let me just make a quick comment for those who have to leave in just a minute. I'm not sure if you know what you've heard. For those of us who have been into psychiatric genetics for many years, 15 years ago thought we were going to find the genes, what you've just heard is pretty much the equivalent of a cruise missile going right into the center of the schema we've had for psychiatric genetics and blowing it up. And it looks like it's an evolutionary approach. It's going to make sense of it eventually. Matt and I are trying to figure out a work group, bringing people here for a weekend to talk about all of this and how evolution could help advance medical genetics. If any of you are into that kind of thing, please send me a note so you can be included in that. Now take questions, please.
Great, thanks, Randy. Yep.
Can you just answer whether what field measured their fitness cost of schizophrenia?
So no, we did that actually in a paper. So you know, you can never measure fitness, right? I mean, you can measure things that you think. Yeah, I mean, you're counting babies, right? But
0.5 for males,
yeah.
0.7 for females.
Yep, or maybe a little lower even for females. I mean, it was lower for males than females, but it was like 0.4 for males, 0.6 for something like that. But this is even counting babies. I mean, it's hard to know. In a modern society, it's hard to know if that, you know, because what's really relevant is the fitness ancestrally. So, other questions, Athena?
How do sort of changes or differences in the amount of inbreeding over historical time influence this story, or do they?
Yeah, that's a good question. So, I mean, one interesting thing about inbreeding is that it's pretty much erased after one generation of random mating, right? So, regardless of how much inbreeding there was historically, it's not going to have any ramification today. That's just it's kind of an interesting principle. But there certainly was a lot more inbreeding historically than there is today.
If there was more inbreeding in the past, presumably there would have been stronger selection.
That's right. Against the recessives.
Yeah.
That's right, exactly. That's actually a good point too. So if you want to purge a population of, say, Drosophila or something of these recessive mutations, how do you do it? You have really strong inbreeding for a long period of time, because then you're exposing those recessive mutations that otherwise are hidden from selection to selection. So, yeah, that's an interesting point I hadn't thought about a lot. So, yeah.
I have a question about how
Please state your questions loudly so everyone.
Oh, I'm supposed to repeat the questions. I'm sorry. Yep.
how was schizophrenia measured?
Okay, so the question is, how was schizophrenia measured? So we're talking about in the PGC sample, for example.
In all your samples.
Well, it's interesting. So there were in the PGC1 there were 17 different samples, and the PGC2 there's like 50. Every one of those does it slightly differently. In some it was clinician diagnosed. In some it was from the IDC-10. But what's interesting is that for some it was simply asking people self-report if they're on psychotropic medications, certain types of psychotropic medications. But what's interesting is that regardless of how it was measured, you get about the same. There was no consistent bias depending on how it was measured. So we were getting the same basic effect size from the samples where it was self-report on whether you were on clozapine or whatever type of psychotropic drug it was, with clinician-diagnosed schizophrenia. So really at this point, I think the PGC's new philosophy is just get as many people as you can, however messily phenotyped. Yeah.
So what does these results say about genetic pleiotropy? Like sort of the axiom is that all these genes affect many different things. You would expect then, so I guess it's sort of the balancing.
Yeah.
Some fairly significant alleles that affect risk to be dragged along because they have some other benefit, but this seems to be saying there isn't any of that.
Okay, so the question is about pleiotropy and psychiatric disorders. There's a great paper by Naomi Ray that uses the PGC data that addresses this question directly using some methods I didn't have time to get to today, mixed linear effect models, treating SNP effects as random effects, and looking at the genetic, this is published in Nature Genetics last year, looking at the genetic correlation between psychiatric traits. Well, first of all, when I said that I think antagonistic pleiotropy isn't a good evolutionary argument, I'm not saying that pleiotropy isn't widespread. I think pleiotropy probably is extremely widespread, but I think it works in the direction opposite of what you'd expect from an antagonistic pleiotropy evolutionary argument, which is to say if it really is antagonistic pleiotropy that's explaining the existence of these alleles, what you'd expect is that there would be a negative correlation between schizophrenia and something or actually, I guess, between schizophrenia and something that's positive, you expect a positive correlation. And what you see instead is a positive correlation between a lot of traits that are negative, which I think is consistent with a mutation selection kind of argument. So you get about a point, it's actually extremely high between bipolar and schizophrenia, which I think a lot of people suspected for a long time. But it's like 0.7 correlation between the two, 70% overlap between the two. And that is true, actually. It is 70% overlap, not 49% overlap. I think it's a common misunderstanding. But yeah, and then for all the disorders, there was a positive correlation, including autism, which is another, I think Bernard Crespi had an evolutionary argument that basically predicted a negative correlation between the two. He predicted a negative correlation. Did he try to explain that?
Tell you more later.
Okay, yeah. Yeah, so the immediate question was about so I'm saying that schizophrenia, we know that schizophrenia is about 80% heritable. We're also saying that the alleles for schizophrenia have been biased towards being recessive. There's kind of a whole host of answers to this question. First of all, just because the alleles are biased towards recessivity, I'm trying to think how best to start this off. Let me start off with the estimate of 80%. Okay, so 80% heritability estimates come from family studies. That's our best guess of what's called a narrow sense heritability, which is the percentage of phenotypic variation that's accounted for by the additive effects of alleles, the additive effects of alleles. Family studies do a horrible job at estimating additive genetic variance. That's well known. So what 80% actually probably is a better estimate of is what's called broad sense heritability, the total genetic broad sense heritability. Broad sense. Yep, broad sense heritability, which is the total genetic variation due to both additive and non-additive genetic variation. So whether or not it's 80% additive or 40% additive, 40% non-additive, we really don't know the answer to that. Okay, so that's one thing. Secondly, if there's a trait that's where the alleles are biased towards being recessive, it isn't the case that you predict that trait is going to have high non-additive genetic variance. It's still completely possible, depending on the allele frequencies, that most of the vast majority of the genetic variation will be additive, will come out additive statistically, even though the mechanism is recessive. Okay? So there's no necessary disconnect between the fact that we're seeing high heritability, even high additive heritability, and that these alleles are biased towards recessive. And I think the third and maybe most important answer to that question is that what we're seeing is a bias towards recessivity. We're not saying that they're all recessive. There's certainly a lot that are dominant. There's a lot that are partially dominant, additive, partially recessive, and fully recessive. There's a whole spectrum of them, right? It's just that there appears to be more on this end than on this end, or at least more on this end than would be expected by chance. Yeah.
So most GWAS studies, 70% don't include the X chromosome, which
Yep.
means on the X are overrepresented for those expressed in the brain.
Yeah.
Did you include the X?
It was included in the PGC, in both of those, actually. There are some hits on the X, but I don't think there are more than
More than you would expect.
would be expected given its size.
And has anyone looked at patterns of X inactivation in XX individuals with schizophrenia?
I'm sure that they have. It's not my area of expertise, but that's an interesting question. Yeah. Do you know anything about that, or?
I just know that there are certain diseases that are at increased risk when X inactivation doesn't work as it should. And so I don't know about with schizophrenia.
Yeah. I don't either.
Dramatic results in Turner syndrome, which haven't been replicated.
Yes.
Huh. Yeah. Yeah.
What was the age range of the reported data?
For people with schizophrenia. So the question was, what is the age range for people with schizophrenia? So I actually don't know.
Are you talking about the onset or?
Well, no, I'm asking in particular.
For the data sets that we used.
Because if it's a heritable trait, it is possible that schizophrenia, being a disease that occurs later on in life after reproducibility, they could be passing it down prior to symptoms.
Okay, so is your question about the sample or about about schizophrenia itself? Because
Age of the sample, I'm sorry.
well, the age of the sample I don't know, and this is kind of a problem with using these huge consortia data sets. We've actually tried to get the age because it's really an important covariate for inbreeding studies. Because it turns out that the older you are, actually exactly what Athena had said, the older you are, the more inbred you are too. So people are getting more and more outbred as time goes on.
The age of onset is, by the age of 20, it's more than half. By the age of 40, it's 50%.
Yeah.
No, I know
Yeah.
that, I was just curious.
Yep. But I think with respect, but I think that the question is relevant with respect to the evolutionary persistence of schizophrenia. I think that's kind of what you had gotten to at the last part of your.
Produced before they're symptomatic, then of course it's less now.
Right, but I think that these things aren't, black and white, right? I mean, there's gradation. And so if you, even if there's a 1% decrement in total number of expected offspring, a 1% decrement in fitness is huge. That thing should be extinct within, 50 generations or something like that. So don't think that saying that, because these are late onset, and this isn't even late onset, it really strikes right during peak reproductive years. But even if something struck as late as, say, the age of 50, right, which is after the vast majority of people have reproduced, if you die at 50, if you have a gene that causes you to become reproductively dead at 50, I'm not saying the body's dead, but you cease to have offspring that gene is going to go extinct very rapidly because there's still enough people, I guess especially men, who reproduce after 50 that is still going to have some kind of negative, even if it's a small effect, some kind of negative effect on fitness. And if there's any negative effect on fitness that's more than, say, 1 in 1,000 or something like that, it's going to be much stronger than drift, right? I think the calculation for it is like something behaves neutrally if it's over about between 1/2 to 1/4 times the population size, effective population size, which in humans is estimated to be about 10,000. So something's going to behave neutrally only if it has a selective coefficient that is less than 1/about 20,000. So if we're talking about something like that is striking at reproductive years or even later in reproductive years, it's certainly a lot more than 1 in 10,000, 1 in 20,000. Yep. Good question.
Figure out the historical recency of some of these SNP alleles, because I mean, if the more recent ones are presumably maybe the ones that not have been kind of selected under pressure, they might be more dominant, and there might be like some that actually fall in that above that heritability curve.
Okay, so the question is about the age of the variants that we've looked at. So no, I don't know a lot about that, but I know some of the broad picture stuff about it. I mean, all the SNPs that we're looking at tend to be fairly old. We know that for a lot of reasons. I mean, one, just if you do some population genetic modeling, you know that things that reach 5 or 10% frequency in the population are going to have an age of, definitely in the hundreds or thousands of generations. That probably more like in the thousands or tens of thousands of generations. Secondly, the vast majority of those SNPs are polymorphic across ethnicities, which suggests they predated the divergence from Africa of about 60,000 to 80,000 years ago. But the stuff that, well, and then there's obviously the case that there's an inverse relationship between the frequency of an allele and how old it is. Okay, so something that's very infrequent is something, they tend to be have a birth date that is much more recent from not long ago. So these CNVs that we were looking at, they have frequencies of like 1 in 10,000. A lot of those are de novo. They arose for the first time in that patient that we saw. Yeah, Athena?
Are there sort of other additional explanations for schizophrenia from either, the perspective of sort of evolution or development that you think have merit and that might interact with what you're suggesting here? Can you sort of give us the broader picture of how you're thinking about?
Well, I think a lot about it with respect to the genetics of schizophrenia. So I don't know if I'm really trying to explain schizophrenia so much as I'm trying to explain the evolution of the alleles that underlie schizophrenia. I think it's really, in a sense, it's kind of impossible to try to explain something like schizophrenia evolutionarily because we don't have a time machine. We can't go back and see whether schizophrenia even existed 1,000 years ago or 10,000 years ago. You know what I mean? So I think the place where I feel like I'm on much firmer ground is trying to understand the evolution of the alleles that predispose to schizophrenia. And there I really do think there's only a few basic types of explanations. I think there's neutral drift, there's balancing selection, and there's mutation selection. And I think a priori, all three of those are, are possible. I think that the weight of evidence to date seems to indicate that mutation selection is explaining most of it. And I say that, and there are some things that I haven't talked about, in this talk that do cause pause. Okay, one of those is that if you look at not the minor allele frequency, but the allele frequency in just raw terms, okay, so the risk allele frequency, what you would expect to see if under purifying selection is something that looks just like what I showed you, and that the stuff that's at 0.9 is going to have even a tiny effect, because things that are rising to frequency, rising to fixation, basically must be behaving neutrally, so it's going to have a tiny effect. What instead you see is something like this, and then you see this tiny uptick over here. It's not probably statistically significant, and I just saw this we just published a paper which had that plot in it, and then I saw this for the first time right after we'd sent it in, and I thought, what crap, I don't know how to explain that. I have no idea what that means. And it's not really consistent with the story we've been saying about schizophrenia. So I don't know. I mean, that's something that has kind of troubled me in the last few months since I've seen it.
Or you could look for antagonist pleiotropy.
Yeah, I mean, so that's the kind of thing that I'm thinking. So I mean, you can't paint with too broad of a brush, I don't think. And I've tried to do that, but I think, and I think that you get some traction doing that. You've gone some way in doing that, but I think that it's very likely that there are some alleles that are under antagonistic pleiotropy, that do have some countervailing benefit, that are behaving more neutrally than you would expect based on simply on their effect on schizophrenia. I don't think that that's going to explain the majority of it, but I do think that might explain some. There might be some that are under frequency-dependent selection. There may be some that are under frequency-dependent selection for some reason completely unrelated to schizophrenia, and they just happen to have some effect on schizophrenia. So I don't think there's a single explanation for this. I think that there's as many explanations as there are alleles, So thousands or tens of thousands of explanations. But I do think that big picture, probably mutation selection is explaining most of it. That's my feeling.
Let's take one or two more questions and stay and talk more with Matt.
Yeah.
Okay, so I want to go back to the phenotype
Yeah.
question, because I work in small-scale societies, and
Mm-hmm.
just took a quick look at a map of the prevalence of schizophrenia across countries of Latin America,
Mhm.
and just eyeballing it,
Mhm.
the countries that have the highest numbers of indigenous groups or individuals have the lowest rates of schizophrenia.
Yeah.
So having done this field work, I'm thinking, okay, how would I even
Yep.
begin to
Yeah.
say that someone has schizophrenia or not,
Yep.
right? And my experience is that some people are off
Yeah.
somehow,
Yeah.
but they're all there are measures, cultural measures to
Yep.
contain them. So what I'm trying to think through rather quickly here is,
Mhm.
in terms of selection acting against a schizophrenic
Yeah.
phenotype.
Yeah.
Under those conditions,
Mm-hmm.
I would think that there would be extremely high selective pressure against those phenotypes
Mhm.
if it involves certain disruption of social relationships.
Yes. Yeah.
Okay, so that's why I'm going back to, what are we talking about?
Yep.
Because you could have a crazy hunter
Yep.
who comes back with a lot of meat but has great relationships with everybody,
Yep.
but does things that are very schizophrenic
Yep.
otherwise.
Yep.
So, those are the kinds of, so the phenotype is actually really important if we're going to start asking questions about the presence of schizophrenia in, our best, model of our ancestors.
Yeah, I think this is a great question, actually. Okay, so I mean, there's so many things I thought of as you were talking about. I should have stopped you, but one of the things that I thought is that it's very hard to compare rates just
Yeah,
baldly
I'm
like
very
that.
aware of that.
There's a great paper by McGrath
Mm.
that really tries to do a good job of doing this, and what he finds is basically that regardless of culture, schizophrenia seems to exist, And it's at that kind of narrow band of about 0.5% to 1%. I mean, if you just look at rates baldly, right, you would think that New Zealand's the most dangerous place to live crime-wise in the world, but it's just because New Zealand does a really good job reporting all the crimes, and that Colombia is a pretty safe place, but it's not, you know. So, it is now.
Yeah.
Is it?
Yeah, it's one of the safest.
There you go. All right. Well, I stand corrected.
Go to Colombia, no,
Venezuela. How about that? Okay.
That's
But I think that the deeper point you're making is really interesting, and that is, you know, something that we don't know much about, and that is what is a selective pressure. I mean, we know we can count babies in Western industrialized countries, and we know what that looks like. It looks pretty bad, and that's mostly due to people having a tough time finding mates. It's not because they're dying at 20 or 30. Although suicide rate with schizophrenia is extremely high, it's not 10% or 20%. But it's mostly because they're having difficulty finding and retaining mates. And so at that level, it's possible that you could see how that would also be the same in any society that you look at. But we don't know. I mean, that's the real issue, is that we really don't know. And that kind of gets back to what I said about Athena, is that we don't have a time machine. We can't go back and look at this phenotype 10,000 or 20,000 years ago. And so I feel more safe saying, well, I don't know. But what I can know, at least have more traction on, is what is the evolutionary cause of these alleles, regardless of whether they caused schizophrenia 10, 20,000 generations ago. You know what I mean? But there was one other thing I wanted to say about what you this is it, is that there is some interesting research that does suggest that in some societies, especially societies that are kind of closer knit, socially less mobile, not necessarily pre-industrialized, but there were some studies in India that were looking at this, where the outcomes for schizophrenia were much, much better than in the West. Are you familiar with this work, Randy? Yeah, and so, that's another kind of thing that causes me pause. I mean, maybe the selection against schizophrenia per se wasn't, I would have a hard time believing there was no selective pressure against a phenotype that looks like schizophrenia in any society, but maybe it was much mitigated, in these kind of smaller scale societies that humans evolved under. I think it's totally possible.
No, let those who want to leave, and anybody who wants to stay for a little private conversation with Matt while he eats his lunch. Thanks, Matt.
Thank you.